Well, good morning or good afternoon, wherever you are, everybody. Happy to welcome you to the Human Islet Research Network, or HIRN webinar series. I'm Joyce Nyland. I'm co-PI of the HIRN Coordinating Center at City of Hope in Los Angeles area. And today we have a talk by Marlie Maestas, who's a PhD candidate in the Washington University School of Medicine, entitled Identification of Unique Cell Type Responses in Pancreatic Islets to Stress. So let me just give you a brief introduction. Wanted to, those of you who aren't too familiar with the network, tell you a little bit about it and how it's transforming, and then introduce today's speaker. So the HIRN, Human Islet Research Network, began in 2014, so we're into our 11th year. It's very fluid and able to evolve to meet emerging science. So it began with the 5 consortia you see on the left: the Consortium for Beta Cell Death and Survival, or CBDS; CMI, the Consortium for Modeling Autoimmune Interactions; CHIB, the Consortium for Human Islet Biomimetics; CTAR for Targeting and Regeneration. HPAC came along later for the Human Pancreas Analysis Consortium that was added. And the HIRN in the middle is as our group at City of Hope. We're the Human Islet Research Enhancement Center to facilitate, coordinate, administer awards for this consortium. The future and merging to the new state is that we still have CBDS and the HPAC, but a couple new consortia are in the process of forming. CMAD, which is the Consortium on Modeling Autoimmune Diseases, came about as a combination of the CHIB and the CMI investigators. PancBase is a new knowledge database consortium that is working to integrate diabetes and pancreas knowledge databases and data from all around the world in the diabetes research community. And the HIRN remains as the coordinating center. We have blank circles there because we haven't come up with logos yet. If any of you are artistic would like to help us. The network now has been going on, as I said, for 11 years, and that's yielded over 1,000 publications by our investigators. 800 researchers have participated over that time, with more than 200 currently involved. That includes over 100 institutions and 12 countries around the world. So it's a very evolving and growing network. We have a HIRN newsletter I want you to be aware of that you could subscribe to. It's for both people inside and outside HIRN. And it will have these webinars announced. It has new members announced and manuscripts. It lists job opportunities in the federal or the private or the university sectors. We list new funding initiatives there, upcoming meetings. And here's an example of a couple funding opportunities you might want to be aware of right now. One is called the New Investigator Gateway Awards for Collaborative T1D Research. And there's the RFA number. This will support a robust pipeline of innovative projects and talented new investigators in T1D research. The awardees' grants will be embedded within our established consortia and that scientific framework. So if you win one of these awards, you become a part of HIRN. And this is a unique opportunity for new and early stage investigators to increase their understanding of questions in the field and to network and establish collaborations to propel their careers forward. This Gateway Award also benefits the network by infusing new ideas, perspectives, and talent into the whole group. The application dates, there's 2 coming up. One will be in June of this year, June 26th, and then another round next year, March 6th, 2026. Foreign applications are not allowed, but otherwise the usual opportunities apply to all U.S. universities. And the other opportunity that is out there right now is innate immunity in type 1 diabetes. This is being offered by the Helmsley Type 1 Diabetes Program. They're accepting at this time concept notes through the end of next month, April 30th, for projects that explore the role of innate immunity in the development of T1D. And the link is listed there. And this will be recorded if you need to go back to it and get the link later on. We also have a website that may be of interest to you. We have the funding opportunities listed there, so check that out at any time for new opportunities, meetings, awards. Our newsletters are there, other announcements. We also have a resources tab that will give you access to various databases that you see here. And in addition, our resource browser, which we've amassed a set of resources that you can access for diabetes research. And you can follow us on all these various social media networks as well as the newsletter. And just to mention again, the resource browser, we now have over 3,322 resources by type. And you can see it includes antibodies, cells, model organisms, data sets, technologies. So check it out. And there's some curated special collections down there at the bottom that you may be interested in using for your research. And as I mentioned, this tutorial and all the past ones are recorded and will be placed on our YouTube channel. So that'll be coming soon if a colleague of yours missed it or you'd like to go back to this at any time. So a little bit about today's webinar then, and then we'll get started. Just a few reminders, if you could note that this is being recorded, as I mentioned. So please mute your audio and turn off your video during the presentation. But there will be a discussion and Q&A that follows. So you can raise your hand and go off mute if you'd like to discuss with Marlie, or put a question in the chat box. So her topics today will be analysis of single cell sequencing of ER and inflammatory primary human cells, unique islet cell type responses identified, exocrine and endocrine tissue that have differential responses to stress, and chromatin accessibility associated with type 1 diabetes, and a key regulator of islet function and stress response, her main emphasis and topic. So let me tell you a little bit about Marlie. Marlie grew up in the Los Alamos, New Mexico, and did her undergraduate degree at New Mexico State University. She's now a PhD candidate in Dr. Jeffrey Millman's lab of developmental regenerative stem cell biology at Washington University in St. Louis. After she graduated from New Mexico, she started her PhD at Washington University in St. Louis in that program of developmental regenerative and stem cell biology. And now she's in her 5th year of her PhD in Dr. Millman's lab. She's been supported by a T32 grant and recently received the David and Deborah Winston Fellowship in Diabetes Research. And as we see here, her research focuses on analyzing single-cell sequencing to understand islet biology. So with that, I will turn it over to Marlie and stop sharing. Marlie, do you want to bring up your slides?
Yes, let's. Okay, how does that look?
Pretty good. And I think that looks fine. Let's give it a try. Thank you.
Okay, so thank you so much for that kind introduction, and I hope you enjoy my presentation today. So I am Marlie Maestas, and I am a 5th year PhD candidate in Jeffrey Millman's lab, and today I will be going over my recent publication in Nature Communications titled The Identification of Unique Cell Type Responses in Pancreatic Islets to Stress. So first, an outline of my discussion today. So we will start with understanding endoplasmic reticulum and inflammatory stress in diabetes, and then go over my single cell sequencing analysis of stressed primary human islets, and then finally how we can use stem cell derived islets to actually understand islet health. So just to start off, we know that diabetes is associated with the pancreas. And within the pancreas, we have these kind of micro tissues that are called the islets of Langerhans. Within these islets, there are multiple different endocrine cell types, including beta, alpha, delta, and pancreatic polypeptide cells. All these cells really work together to regulate our blood glucose levels. However, in diabetic patients, this goes awry. So we see that within patients, they have death or dysfunction of these pancreatic beta cells. And the pancreatic beta cells are important because they actually produce insulin. And this insulin biosynthesis begins with transcription of the insulin gene within the nucleus, and then translation of this mRNA into pre-pro-insulin, which occurs within the ER. Upon translocation into the ER, this signaling peptide is removed and it creates proinsulin, which then goes through the Golgi and is folded within the ER and is stabilized with disulfide bonds. It then is transported to the Golgi and into granules, where the proinsulin is cleaved into C-peptide and insulin, where then can be secreted from the cell. However, one problem is when this insulin is being properly folded, it can actually become misfolded or unfolded. And when that happens, this can cause increases in endoplasmic reticulum stress. And this ER stress can eventually lead to the unfolded protein response pathway, and this will either lead to anti-apoptotic responses and homeostasis of the cell, or it can actually lead to pro-apoptotic outputs that cause apoptosis of the cell, like we can see in diabetes. Now, unfolded protein is not the only way that this ER stress can occur. It can occur through overload of protein, actually mutations in the protein, environmental toxins, viral infections, as well as aging. In my talk today, I will be focusing on how ER stress and inflammatory stress affect primary islets. And so they've seen that with sections of primary islets from patients, so here we have adult control cells, as well as type 1 diabetic cells, and then pediatric control. And in this graph, these authors are looking at CHOP immunostaining. And CHOP is a transcription factor that is upregulated in response to ER stress. So we can see within type 1 diabetic patient, they have increased expression of CHOP, indicating that these cells are going through ER stress. And when this stress is severe or prolonged, it can lead to programmed cell death. With this bottom graph, we're looking at BIP expression. So BIP is a chaperone protein located in the ER, and it actually assists with proper folding of newly synthesized proteins. So it helps prevent the accumulation of misfolded proteins. And we can see a slight increase within our T1D patients. Another interesting finding from previously published data is showing that as beta cell markers go down, we actually see an increase in that chaperone protein in the ER. So kind of indicating that beta cells can lose their identity as they go through this ER stress. So my main goal is really to identify genetic regulators of pancreatic islet stress response. And we really want to do this using a single cell method to understand the differences in how different cell types within the islet actually respond to stress. So our experimental plan is we took primary human islets and we applied a variety of different ER or inflammatory stressors. So we have DMSO as our control, and then Burfeldin A. So Burfeldin A acts as a protein transport inhibitor. So we'll actually stop the transport of protein from the ER to the Golgi, and this will actually cause both ER and Golgi stress. Then we have Thapsigargan. This inhibits the SARCA2B pump. So this causes a depletion of ER calcium stores, leading to that ER stress. And then finally, we have different combinations of pro-inflammatory cytokines, such as IL-1 beta, interferon gamma, and TNF alpha. So we have one that has all three, which I will refer to as cytokine mix, and then we have different combinations of them. So what we can do is we treat our primary human islets with these different stressors. Then we will actually incubate them in a hashed antibody that will bind to the surface of the cells. And this allows us to pool all of our samples together and then do a single cell RNA sequencing run. And during analysis, we can deconvolute these samples to identify which cells were treated with each stressor. So we did this across 5 different patients. So we have 5 patient analysis, which range from males and females as well as different ethnicities. So here we wanted to really look at if our 5 patients respond to stress similarly. Now it's already known that primary islets can be very heterogeneous in their response to different stimuli, as well as just their gene expression or function in general. So here, yes? Oh, sorry. So here we looked at top markers for each stressor and their gene expression across patients. And you can kind of see overall that each patient does respond similar to our stressors. So next we actually combined our patients to make a UMAP to identify the different cell types within our population. So here we can see that we have 4 subpopulations of alpha cells, as well as 3 populations of beta cells, and then other endocrine and exocrine cell types. And we do this identification based on top gene expression markers here in the middle heat map. Like I said earlier, because of the hashing, we can identify which stress was applied to each cell. And so this is a UMAP, but instead of looking at cell types, we are now looking at the different stressors. So interestingly, you can kind of see that different subpopulations kind of arose because of the stressor that was applied to them. So for example, if we look at our alpha cell 3 population, you can see that it's almost completely comprised of interfering gamma cells. And we can also see with our beta cell 3 population as well as our alpha cell 4 population, these are almost completely comprised of BFA treated cells. So we also wanted to just make sure, are our stressors working as they should? So we looked at top markers that we would expect to be expressed under certain conditions. So at the top we have STAT1 and CXCL10, which you can see are highly expressed within our cytokine mix and different cytokines. Then we have HSPA5 as well as HERPUD1, which is highly expressed within our BFA treated cells. Then we also have MANF and SDF2L1, which is more highly expressed in BFA as well as dapsogardin. So from this, we just confirmed that our stressors are working as expected. So to look deeper into what's happening when we stress these cells is we looked at the number of differentially expressed genes across cell types and across stressors. So what we're seeing is each of these dots is the color indicates the type of stressor applied. So for example, these top dots, which are purple, indicate that they were treated with BFA stress. Then above 0 is the number of differentially expressed upregulated genes between that stressor and control. If it's below 0, these are the number of downregulated genes. And we can kind of see right away that the beta, alpha, and ductal cells seem to have the most robust response to these ER and inflammatory stressors. We can also see that BFA, like I pointed out earlier, these purple dots, and cytokine mix, this yellow, have induced the strongest response across cell types. So this is really an overview, but we really wanted to look deeper. And as I mentioned earlier, one of the responses from ER stress is the unfolded protein response pathway. So this pathway has three main sections, and one of the sections is ATF6. So this is an ER membrane-bound transcription factor, and it is triggered by that accumulation of protein. When it senses that, it translocates into the Golgi, where it is then cleaved and releases a fragment that moves to the nucleus to then activate these UPR-associated genes. The second section is PERK. So PERK will autophosphorylate, and then it works in conjunction with eukaryotic translation initiation factor 2 to actually stop translation. So this phosphorylation of ELF2 alpha reduces the start codon recognition, which slows down translation, which this then allows the induction of ATF4, which is a transcription factor, and this will induce XBP1 and CHOP and GATA3, which can lead to cell death. And then finally, we have IRE1. So this senses unfolded protein and then it actually dimerizes. So this has a endonucleose domain and a kinase domain. The endonucleose domain will target XBP1 mRNA and splice it, causing XBP1 spliced. And this transcription factor regulates both ER-associated degradation, so ERAD proteins, as well as chaperones. And it can actually lead to degrading of these misfolded proteins. So we wanted to look at these three sections within our data set. So I know this is a lot of data to look at, but overall what's happening is we looked at our major endocrine cell types, so alpha, beta, delta, and PP cells, and then the exocrine cells, so ductal and acinar, across different stressors. And then we looked at our three sections of the UPR and then also ERAD. And then along this axis is the different genes associated with these pathways. So just to point out some interesting things from this, within BFA, IL-1 beta, and interferon gamma, and TNF alpha cells, we see that DDIT3 is highly expressed within the beta cells. And this is a gene that is indicative of apoptosis. We also see that the highest expression of anti-apoptotic genes, such as BIRC2 and AP15, is actually more highly expressed in the alpha cells under BFA, IL-1 beta plus interferon gamma, and interferon gamma cells, more highly expressed in the alpha cells rather than our beta cells. We also see that the exocrine cells, so acinar and ductal cells, seem to show high expression of PERK-associated genes compared to endocrine cells. However, we also see that the endocrine cells actually have strong expression of genes associated with ATF6. So we're really seeing differences not only across cell types, but also across tissue types. So we wanted to investigate this a little further. So what we did is we took our tissue-specific differences. We looked at endocrine cells as a whole. So we took our alpha, PP, delta, and beta cells, and we subsetted those. And then we also took exocrine cells, so acinar and ductal cells. And then we wanted to compare these two tissue types. So if we first just look at the number of differentially expressed genes between these tissue types, if we first look at BFA versus control, we can see that BFA actually induces a higher upregulation of genes in our endocrine cells compared to our exocrine cells. However, if we look at the number of downregulated genes, there's actually more downregulated genes across exocrine cell types rather than endocrine cell types. When we look at thapsigargan, we kind of see a different effect where the exocrine cells are having more upregulated genes under thapsigargan stress, and then the downregulated genes are pretty even between the two cell types. And then finally, in cytokine mixed stressed cells, we see that treating the cells kind of affects both tissue types similarly in their number of differentially expressed genes. If we look more closely at what pathways these genes are associated with, we can really see major systems that are being differentially regulated between tissue types. So I'm just going to point out a couple interesting things. So within our endocrine cells under BFA stress, we see that both autophagy and the ciliary landscape are both upregulated in these endocrine cells. While in exocrine tissue, glucokinase activity is actually being upregulated. In fats of organ stress, because we see the most within the exocrine tissue, we also see them associated, these genes associated with ferroptosis, which is a type of cell death, and then also cellular response to stress. And then finally, when we look at our cytokine mix treated cells, we see that this induces an upregulation of programmed cell death within our endocrine cells. And in the exocrine tissue, we see PD-1 signaling as well as MHC class II antigen presentation. So these are just some of the different pathways we see being differentially expressed. Not shown here, but we do see upregulation of T1D associated genes across cytokine mix stressed cells. So one of the things we thought was interesting was this programmed cell death under cytokine mix. So we kind of wanted to understand if the cells could they recover after being treated with cytokines. So here what we did is we took primary human islets, we treated them with cytokines for 48 hours, and then we actually removed the cytokines for an additional 48 hours. And what you can see is we have high expression of genes that are expected to be expressed under cytokine mix, such as MT2A, CXCL11, CXCL9, IL-32, and SOD2. So we can see that these are highly expressed in our cytokine mix treated cells. However, when we actually remove the cytokines, this drops dramatically, almost to basal levels in many of the conditions. And then we also see that we have a reduction in MAF-A, which is a maturation marker in islets. So we see a reduction under cytokine mix. However, this does start to come up if we remove the cytokines. So this data together really indicates that these islets, these human islets, can recover after cytokine stress, even with these significant transcriptional changes during inflammation. So the next thing we were interested with the cytokines in particular is investigating the chromatin changes. So we conducted single nucleus multi-omic sequencing of cytokine mix treated primary islets. Just to remind you, cytokine mix is a combination of IL-1 beta, interferon gamma, and TNF alpha. So here, if we look at this top UMAP, this is identifying the cells by their treatment. We can see that within the RNA population, so this is only using RNA data to determine cell types, that it's more heterogeneous and there's kind of a mix between cytokine-treated cells and PBS. Whereas if we look at just our ATAC data, they almost cluster separately. And when we combine these two together, we still see that distinction between cytokine mix treated cells and PBS treated cells. So we also looked at genes that we had already identified in our single cell sequencing data. So we looked at CXCL10, SOD2, IL-32, GBP4, and LY6E. And we first looked at their gene expression. So as we expected from our other data, this was highly expressed within our cytokine-treated cells. Then we wanted to look at the chromatin regions associated with this gene, and you can see that they are more accessible within those regions as well, which makes sense. So going kind of back to our interest in these tissue-specific differences between endocrine and exocrine cell types, we looked at the motif accessibility across these cell types. So what we did is we took individual cell types. So if we look just at our beta cells, I took just the beta cells and I looked at the top 20 differentially accessible transcription factor binding motifs between cytokine mix and control. So that would be these top 20 motifs. Then I made a heat map and looked at the accessibility across our 5 different cell types. And I did this with all 5 cells. So interestingly, between beta, alpha, and delta cells, there was actually only 3 motifs that were the same across their top 20 motifs. So this was FOSL1 June D, and then RBPJ, which is notch signaling, as well as ZNF282. So there's only 3 shared between beta, alpha, and delta cells. However, when I looked at the exocrine cell types, they share 15 common motifs in their top 20. And then we can also kind of appreciate that across the endocrine graphs, you see more kind of ubiquitous upregulation of these motif accessibilities. So you can see the highest expression within that cell type of interest. However, you do still see accessibility in the other cell types. But if we look at our exocrine graphs, these are really more specific to that tissue type. And then finally, there was a paper that came out that found that reductions in the enhancer that contain a specific risk variant for type 1 diabetes was also associated with reductions in CFTR expression in ductal cells. And so we kind of wanted to see, can this also happen within our data set? So we looked at this risk variant, so RS7795896, and we see that chromatin accessibility is reduced with the treatment of cytokine mix in our ductal and as well as our acinar cells. And then we also see reductions in CFTR accessibility and gene expression. So I think this is really interesting because this data shows that environmental factors such as adding cytokines or inflammation cause similar transcriptional changes in ductal cells to what we see in type 1 diabetes patients. So after exploring these different tissue-specific differences, we wanted to look closer at the endocrine cell types across stressors. So we kind of went back to our single cell data and looked at gene expression across our different stressors and within beta, alpha, and delta cells. So just quickly to look over this, overall we found under stress conditions that cell types show increases in genes associated with ER stress and or inflammation markers, as well as decreases in islet identity markers. And like I showed at the beginning where that paper had decreases in beta cell identity while BIP was increasing, we see a similar trend within our data set. The neat thing about this data set is you can look at each individual cell type, you can see what they have in common and what they have different. So the top is looking at Venn diagrams and number of differentially expressed genes within beta cells specifically. There would be 605, whereas beta and alpha cells under BFA stress have over 2,000 genes that are differentially expressed. Through this data, you can also identify cell type specific markers. So for example, within BFA stress under beta cells, you can see CRELD2 is upregulated, as well as a variety of other genes. And so this data I'm showing right now is our 3 major stressors, BFA, Thapsogardate, and cytokine mix. I did do this with all the individual cytokines, and some interesting things from the individual cytokines is looking at genes associated with antigen processing and cross-presentation, as well as interfering gamma signaling. So let's first look at this heat map on the left. So this far heat map is cells, beta, alpha, and delta cells under control conditions. And then on the right is these cells treated with IL-1 beta and interfering gamma. So if we look at our control cells, we see that mostly across the genes associated with antigen processing and cross-presentation are expressed within the alpha cells in basal conditions. But what's interesting is once you add these cytokines, these markers shift to being more expressed within our beta cells. And specifically, there's genes associated with CD8 T cells to promote their infiltration that are being expressed in beta cells after the addition of cytokines. And we see a similar trend within interferon gamma signaling. So under basal conditions, our alpha cells are mostly expressing these genes associated with this pathway. However, when we actually add interferon gamma, we see a more heterogeneous expression, where now some beta cells, some alpha cells, some delta cells are expressing these markers. So finally, we actually wanted to look at just the beta cells themselves because of their important role during diabetes. So what we did is we took our single cell sequencing data and we subsetted just the beta cells. And if we first look at beta cell identity across different stressors, we find that BFA induces this, down regulation of beta cell identity genes. Cytokine mix does this as well. And then different genes are differentially expressed depending on what stressor was applied to them. So one of the things we wanted to look at was chromatin remodelers. So all these different genes across, I've only pointed out some of them, but all these little squares are a single gene. And looking at INO80, CHD, ISWI, BAF, and PBAF. And we can see that BFA again induces this increase in genes associated with chromatin remodelers, which made us interested in understanding the chromatin that occurs when we add BFA to the cells. So again, we did multi-omic sequencing of BFA and DMSO treated cells. And one of the first things we found was under BFA stress, insulin within our beta cells is less accessible than our control cells, where we see the opposite with ER stress associated protein HSPA5. So this gene is actually more accessible when we add BFA to the cells compared to control. Now, one of the most interesting findings from our chromatin data within BFA treated cells is we compared the enriched motifs, so transcription factor binding sites, and upregulated genes. So we have our motifs and our RNA that is being upregulated under BFA stress in beta cells. So we found 43 that were overlapping. But then we wanted to know, okay, we can use this data to actually compare to other cell types to identify a beta cell specific changes. So we compared these 43 RNA and motifs that overlap to other endocrine cell types, so alpha, delta, and PP cells. And we found that there's only 5 that are actually unique to the beta cells. So this is BOC2, which has actually been shown to be a risk gene for type 1 diabetes. We have SMAD5, which is a protein downstream of TGF-beta signaling and can promote glycolysis. We have HMBOX1, a regulator of telomerase activity, TP53, a gene that's also been connected to T1D and T2D by TP53 mediated apoptosis. So that was really interesting because we saw this gene only being upregulated within the beta cells. And then JUND is a gene associated with beta cell dysfunction. So I know I've shown you a lot of sequencing data so far, and one of the things we really wanted to do was, can we actually use this data to really investigate islet health in vitro? So the first thing we needed to do was decide on candidate genes to investigate. So we found that there's actually only 6 genes that are upregulated across our different stressors, so cytokine mix, thapsigargan, and BFA. So different types of ER and inflammatory stress are only upregulating 6 of the same genes within alpha, beta, and delta cells. So these genes are CYP1, ERP44, which is involved in protein folding, HSP90B1, which is a chaperone protein in the ER, NEAT1, a long non-coding RNA, SELC, which is a transmembrane protein in the ER, and then VMP1, protein involved in autophagy. So a lot of these genes were kind of expected, I would say. However, CYP1 was really unknown. So CYP1 is a calcium and integrin binding protein 1. So in other cell types, it has a multitude of functions. And we really wanted to understand, how could CYP1 be playing a role in islet health? So here, if we look at CYP1, it can play a role in adhesion, survival, proliferation, calcium flux, gene transcription, which, as we know, a lot of these are, or all of these are very important for beta cell function as well. So to test CYP1, we used our stem cell derived islets. So we have different iterations of this protocol have been published over the years, and our most recent one can be found in our Nature Protocols paper that was published in 2021, as well as a Nature Biotech paper in 2020. So our differentiation has 6 stages, and after these 6 stages, these cells will eventually produce glucose-responsive cells that secrete insulin and express all the major markers of islets. So here, this is a glucose-stimulated insulin secretion, showing that our stem cell-derived islets at a low glucose respond, as well as respond more or higher in a high glucose environment. We also show that our islets will express the major markers of primary islets, such as C-peptide, glucagon, somatostatin, NKX61, so on and so forth. So these cells really make for a great model and are able to be genetically modified to study islet health. First thing we needed to confirm is, do our cells actually respond to our stressors? So here we applied cytokine mix, dapsgargan, and BFA. We see that caspase-3-7 is increased under dapsgargan and BFA stress. We also see with islet identity genes, insulin is decreased with certain stressors, as well as glucagon, which is what we see within our primary islets. And then we also see increases in ER stress markers. So confirming that these stem cell derived islets do respond to stress and would make a good model system. So to study CYP1, what we did is we wanted to modulate it in our stem cell derived islets. So we transduced them with lentivirus, either carrying a short hairpin RNA to reduce the expression of CYP1, or an open breeding frame to actually overexpress this gene. So here are just gene expression of CYP1, whether it's a knockdown or overexpression. And we can see with knockdown, we get reductions over 50%, as well as overexpression is much higher than at basal levels. Now, if we look at the function of stem cell-derived islets with knockdown of CYP1, we actually see this results in a significant increase in insulin secretion under both 2 millimolars of glucose as well as 20. But at the same time, if we overexpress CYP1, we see no differences in function. Finally, we looked at CYP1 and its role during stress response. So here we just looked at caspase-3-7, and we had CYP1 knockdown and CYP1 overexpression. When we knock down CYP1, we actually see increases in apoptosis via caspase-3-7 under BFA, cytokine mix, and dapsgargan stress. So all 3 stressors, when we knock down CYP1, they have more apoptosis. However, when we overexpress it, there's no significant difference in cytokine mix and dapsgargan. However, in BFA stress, we do see a slight reduction in apoptosis when we overexpress CYP1. Finally, we wanted to see if we could actually identify drug targets instead of specific genes. So we used this drug gene interactive database to identify drugs that could target genes of interest instead of genetically modulating them. So we have RELA, which is a subunit of NF-kappa B, which in this case is cynaropyrin, targets RELA, and then CKB, which is an enzyme transfer phosphatase, and this is targeted by licoritin. And then we have P4HB, which is an oxyreductase complex, and this is going to be targeted by acyclovir, which actually has been previously shown to reduce insulin resistance in mouse models. So we found these 3 genes because they were all upregulated in under BFA stress in beta and alpha cells. So again, we took our stem cell-derived islets, we applied stress for 24 hours, and then we applied our drug for an additional 24 hours. And what we see is with the addition of the drug, we have reductions in caspase-3 expression. Whether it's the genes that target RELA, CKB, or 4-HB, each of them slightly reduce apoptosis within our cells. So in conclusion, I've made a comprehensive index of transcriptional changes that are driven by this ER and inflammatory stress in pancreatic human islets. And we really leverage this single cell technology to identify cell type specific and tissue specific transcriptional responses. And then our ATAC data really suggests that microenvironmental cues like stress could play a similar role in T1D to that of risk variants. Finally, we identify these 5 transcription factors that are very specific to beta cells, which could be further investigated to understand beta cell health. And then finally, we use our stem cell-derived islets to modulate gene expression to really understand islet health and if we can improve their response to stress. And here's my acknowledgements. Thank you so much to Jeff for helping me with everything and being a great PI. So yeah, thank you everyone else that helped me with my paper as well. And that's it.
Great, thank you, Marlie. That's a very interesting talk. Appreciate it. We have one question in the chat so far from Alexandra. Have you looked at single cell analysis data of isolated islets or pancreas samples from recent onset T1D subjects to check the correlation with your data? What ER stress markers are upregulated in C2 and beta cells or other endocrine cells? The main question is how closely data from in vitro ER stress stimulation experiments correlate with what is happening in the T1D pancreas. I tackled that one. And Alexandra, if you'd like to go off mute and interact, that would be great too.
Yeah, so we actually did use some published T1D data sets, and I compared this to my cytokine treated cells. And we actually saw if we look at upregulated pathways between type 1 diabetes and cytokines, we see a lot of overlap. I didn't specifically look at all different, ER stress genes and UPR associated genes, but we did see an overlap in upregulated pathways between these 2 data sets. And I think that will become even more available, like with PancBase and all these new databases that have so many different patients, that will make that kind of analysis much easier.
That's true. That'll be a big benefit to having PancBase. Alexandra, did that answer your question?
Yes, thank you so much for your answer. I just heard different opinion about ER stress in type 1D pancreas, and some opinions believe that it's not ER stress ectopic, but synthesis of proteins decrease, but but ER stress markers will not like upregulated. There is another opinion. There is another way not not connected with ER stress, and I try to have like to try to have big picture of it, like not focus only ER stress, what else or what apart from it could happening if we not see upregulation of that markers. What about it? Like, what do you think? It's every time like ER stress upregulation in recent onset type 1D, or it's could be another like processes of processes which contribute to decrease of protein synthesis.
Yeah, I mean, I think T1D is such a heterogeneous disease, so it can really work through so many different pathways and methods. I do think ER stress is a big factor, but I agree that there are other ways that the protein synthesis could be disturbed.
Thank you so much.
It is very complex.
Great work.
Thank you. Did anyone else have any questions for Marlie?
Hi,
Yes.
George. Yes, this is Infan from City of Hope.
Yeah.
And Marlie, that's a great talk. And I recently started some single nuclear RNA-seq study for islets, and I just have some questions. Curious about your single cell sequencing. I know you use the immunostaining as a way to reclustering them, but still sounds very astonishing to me. Your treatment can introduce completely different subcluster of beta cell because you do have a control group, and you say the control sample only contribute to one, say the beta cell category, right? Is the subtype 1 or 2? So how do you think about that? Like for me, if we use rodent islets and increase one ER stress transducer overexpression, we see dramatic phenotype, but when we comes down to the subcategory, we don't see any new subcategory of beta cell to, we can't observe a new subcluster. But here you obviously see very dramatic difference, which can give you a different subcluster. Do you have any thought about that?
I mean, I think in different cases, it could depend on concentration of your compounds as well as timing. So these are, on the higher end of the concentration. So maybe it's inducing a larger transcriptional change, which allows the kind of subpopulations to arise. I will say throughout my manuscript, I really don't focus on the subpopulations. I really focus on cell types and then what stressor was applied to them. There are some interesting differences you can see, but yeah, we don't mostly focus on those being subclusters.
Okay, thank you.
Sure.
Thank you. Any other questions for Marlie? Okay. Well, thank you all for attending, and keep an eye out for our next HIRN webinar. Should be in another month or so. And do sign up for the newsletter and keep posted that way too. Thank you, Marlie, for an excellent talk.
Thank you.
Time today. Thank you.
Thank you so much.
And I hope to see you all soon.